Alright guys, so here we are, a different shirt, a different place, but the same idea. Let's continue talking about bioenergetics. I know you love it, I love it too. Let's be real. Guys, what are we talking about today? Fermentation, okay? This is one of those things that when humans start understanding how it works, it literally changed everything. And I also give the IMAT an excuse to start asking about this in the IMAT. We're also going to talk about the link reaction. What happened when we have the pyruvate and we don't have to do fermentation, okay? What is the next step in this pathway of the cellular respiration? Finally, we can't leave this aside. We're going to be talking about some of the questions that the IMAT has asked about this topic, especially 2025, the last edition, because we're going to see so many examples of it, so it's good for you to be prepared. So let's just jump straight to it. Okay guys, so I just want to refresh your minds about the glycolysis because it's going to be important. But first of all, if you didn't watch the first video of bioenergetics, please go jump straight to that video because it's going to be really important for us to build this ladder and for things to make sense. So the only thing I want you to keep in mind of the glycolysis is that we're going to start with glucose and we're going to end up with pyruvate, okay? And usually this is split into two different phases. The first phase we're going to call the investment phase. We're going to literally invest 2 molecules of ATP, as you can see it right here and right here. But no worries, because after that we're going to have the payoff phase. As you can see here, we're going to get those 2 ATP back here, and also an extra 2 ATP as a net gain, okay? Because remember, this is just half of the molecule of glucose, so this is going to happen twice, giving us those extra 2 ATP. Last video, I gave you a lot of details of this process so you can just stand out and be ready to answer any question of glycolysis. But if there's something that you can't forget in no possible way, is this mantra, okay? 2, 2, 2. This is what you're gonna have at the end of the glycolysis. So 2 molecules of pyruvate, 2 molecules of ATP as a net gain, and 2 molecules of NADH. Remember these guys? These are gonna be those cofactors responsible for transporting electrons straight to the mitochondria to produce ATP. So they're not enzymes, but they're very important because they're gonna be part of the process of oxidizing the substrate, the glucose. Okay, so this is where things start getting interesting. We did it, we made pyruvate, we have a bunch of them. What is the next step? The logical thing to do is let's move on and let's get into mitochondria, because it's there that we're going to produce a lot of ATP. But it's not always like that. When we don't have oxygen, something different is going to happen. So we're going to split this fate of the pyruvate into two. An aerobic pathway, that's where oxygen is present. So the pyruvate is going to move into the mitochondrial matrix, and we're going to start the process of pyruvate oxidation and the Krebs cycle. And when we don't have oxygen, the anaerobic pathway, okay, this is going to lead us to the fermentation process. So the pyruvate is going to still stay in the cytosol, and all the fermentation is going to happen in the cytosol. Let's take a look at this more carefully. Okay, so I know you are familiar with the fermentation. You have heard about it whenever you're talking about wines, beers, and other beverages. Fermentation is right there, also in the bakery store. But I want to bring you the chemical point of view. The fermentation is just a solution, a way out, a plan B. Because whenever we have pyruvate, the idea is let's move on to the mitochondria to produce a lot of ATP. But if there's not oxygen available, it requires oxygen. Remember, the oxygen is going to be the final acceptor of electrons. But if there's no oxygen, we have to find a way out. So the fermentation itself does not produce any energy at all. It's just a plan B. Usually you might see some materials associating fermentation as the productions of 2 ATP. This is very common, unfortunately. They're just mixing the two concepts of glycolysis and fermentations because they kind of happen together whenever the cells realize there's no oxygen present. But the 2 ATP is not coming from the fermentation, but from the glycolysis, as you know. So what is the purpose of the fermentation? Why is this happening? Okay, I understand there's no oxygen available, but why do I have to do that? Because I already have those 2 ATPs, right? I don't have to do anything if it doesn't produce energy. The thing is, remember I mentioned that one of the byproducts of the glycolysis is the formations of 2 NADH. And this 2 NADH, they are cofactors responsible for transporting electrons to the mitochondria. But if they can't go there, they're just going to be hanging around saying, okay, what do I do now? I have these electrons with me. I am a transporter, but I can't transport anymore because I'm taken. And the fermentation is going to be the guy who's going to sort that problem. Because believe it or not, we do not have much of this NAD in our system. There's a limited amount, and if they're taken, unfortunately we can't go back and do more glycolysis. So if we can't produce energy in the mitochondria and we cannot produce energy through glycolysis because the NAD are taken, that's a problem. That's a huge problem, and we don't want that. So we're gonna have our plan B here. The NAD will be generated from NADH. We're gonna go back to NAD+, the form without electrons, and this is going to be very important so we can go to another round of glycolysis and produce an extra 2 ATP. I know it's not a lot, but it's better than nothing, right? We're gonna have two different types of fermentations. They're both are gonna take place in the cytosol of the cell, but they are going to happen in different organisms. So we're gonna have the lactic acid fermentation, usually the one that is most asked because it happens in human muscle cells. Not only that, it also happens in the blood cells, also happen in our liver and in some bacteria as well. And we're gonna have the alcohol fermentation. That's the one that people usually think the most. That's where we're gonna produce alcohol, obviously. And it's gonna happen in yeast mostly, and some type of plants. Remember, yeast, it's a type of fungus that is basically built by just one cell. And the first step is going to be equal for both of them. We're going to start with the pyruvate, and the pyruvate is going to be the one who's going to receive the electrons that were present in NADH, so it can go back to the form of NAD+. Once it's received, it's going to be converted into something else. Here in the lactic acid fermentation, it's going to be a straightforward process. Once it receives those electrons, it's going to chemical react and convert into the final product that's going to be the lactic acid. That is the one that is going to give you the pain whenever you're hitting the gym. So one of the ways you can get rid of that is just massaging, so you're just spraying that lactic acid. On the other hand, we're going to have the alcohol fermentation. It usually takes 2 steps, but it's not important for you to remember that, but it's just a little bit longer. We're going to have an intermediate ingredient here. Whenever the pyruvate receives those electrons, we're going to have the acetylaldehyde, and then finally we're going to have the ethanol. So the byproducts of the alcohol fermentation is going to be the ethanol and the CO2. I mentioned that it is responsible for making bread. What about the alcohol? Why don't I get drunk whenever I'm eating a piece of bread? Well, that's a good question, actually, something that I asked myself for a long time. But whenever you're baking the bread, it's going to produce the alcohol because it has a fermentation process going on, but the ethanol will just be evaporated with those high heats in the oven, okay? And the only thing that's gonna rest there is gonna be the CO2, the one responsible to make the dough grow. Science is beautiful. 2022, very quick question here. So this is basically asking us, where does the 2 different types of fermentation takes place in the cells? So in the first row here, we're gonna have the alcohol fermentation in yeast. In the second row, we're gonna have the lactic acid fermentations. So each row, we're gonna have different options, but if you're very sharp in fermentation, you're going to remember that it doesn't really matter. They both take place in the cytoplasm. The only option that gives us this is going to be row 3, leaving us with option letter A. 2014, this is an interesting question here. It has a little bit of mysterious. It tells us the diagram shows the respiration process in the cytoplasm of a yeast cell or an animal cell when O2 is limited. Oh, wait a minute, O2 is limited, so we're talking about fermentation. Okay, we are starting with glucose. Something is transformed, and from this we have a Mysteria X product. Okay, what are the conditions? O2 is limited in a respiring animal cell. Okay, so animal cell. We know that we're talking about fermentation. This can only be lactic acid. Okay, and the other side, we're gonna have this taking place in a yeast cell. So the Z can only be an alcohol. And what about the Y? What is it? By the context here, it can only be pyruvate. The only option that give us this, let's take a look. Let's focus at the Y first. Pyruvate, and pyruvate can only be one of these 2. X, we said it was what? Lactic acid, okay? So it can only be letter E. So yeah, guys, fermentation to the IMAT is pretty much that. I need you to remember that it does not produce any ATP. The purpose is to regenerate NAD+. It takes place in the cytosol. And for you to stand out and, be the difference here, just remember that if the electrons from the NADH is leaving these cofactor and go into the pyruvate, who is oxidizing and who is reducing. This is important information you can't forget. So the electrons is going to the pyruvate, so if it's receiving the electrons here, the pyruvate is reducing, and an NADH is oxidizing. This is a very clear type of questions from the IMAT, so it's important for you to always keep in mind who's reducing and who is oxidizing. If this is not really clear to you, I talked about this in the previous video, so once again, go back there, just revisit, and if you have any questions, just leave a comment and I'll be happy to help. Okay, I just turned on the light because it's starting to get really dark in here. Let's talk about whenever we have enough oxygen to our process, okay? We're talking about cellular respiration, we're talking about pyruvate entering the mitochondria. Because true thing is, the glycolysis is the only step that happens in the cytosol. After that, every single process is going to happen in the mitochondria. The idea here is to produce a lot of energy, around 30 to 32 ATP. We're going to see how we're going to get this. And as other byproducts, we're going to have the CO2 and the water. Just a little throwback about the mitochondria. Remember, this is an important organelle that has some resemblance with bacteria. For that reason, it has its own DNA. It's going to have two membranes, one outer membrane and one inner membrane. As you can see here, it's kind of a little bit wrinkled. So we're going to have these cristae here. That is going to be important to have more area of contact, because right here we're going to have important proteins that are going to be responsible for the oxidative phosphorylation. That's the final step in our cell respiration. And we're also going to have all the important proteins for the Krebs cycle here in the matrix of the mitochondria. So, okay, the link reaction, that little process that happens before the Krebs and after the glycolysis. It's also called the oxidative discarboxylation. Oh, that's an ugly name, but it really helps us to understand what's going to happen here. So it's going to oxidative because it's going to lose electrons, and discarboxylation because, well, guess what? It's going to lose a carbon. First things first, we're going to have the pyruvate that we produce in the glycolysis, and it has to enter the mitochondria. We're going to have some transport proteins responsible just for that. Not important for us to remember the name, but once it's in, it's going to go straight to the matrix, and there is going to happen the first reaction inside of the mitochondria. I'm going to be honest with you guys here. The important thing to remember and to keep in mind and to visualize is that the pyruvate has 3 carbons, as you can see here. At the end of this link reaction, we're going to end up with 2 carbons. I know it seems a little bit silly, but sometimes the IMAT likes to ask you how many carbons are generated after a particular process. Okay, so like for example, asking how many carbons that we're gonna have after the Krebs cycles, like CO2 being produced as a byproduct. So you might think, oh, 3 because the pyruvate has 3 carbons. Wrong. 2 because we're gonna have this link reactions happen before the Krebs cycle. And how many carbons we have here? 2 carbons. So once this enter the Krebs cycle, that's gonna be our next step. These 2 carbons are gonna be released as CO2. It's important for us to keep in mind these molecules, but we're gonna have the pyruvate, and this is going to be converted into something very important and that I'm sure you heard about, that is going to be the acetyl-CoA. For this to happen, we're going to oxidate this molecule, and if we're oxidizing, we're removing electrons from this subtract, okay, from this molecule of pyruvate, and by doing that, somebody has to receive it. It always happens in pairs, remember? So who's going to be responsible for receiving and transporting these electrons? Well, that's no mystery, it's going to be the NADH. And if we have 2 carbons here and 3 here, that means that 1 had to leave and it left as a CO2 form. So there's no big mystery here. Basically what's going to happen is that at the end of this process, we're going to have 2 molecules of acetyl-CoA per molecule of glucose. We're also going to have 2 molecules of NADH per molecule of glucose and 2 molecules of CO2 per molecule of glucose. Remembering this and having very clear in your mind what is coming in and what is leaving, you're really fine with the link reaction. Okay, so if you're with me until this point, you are a warrior. Let's go to the final topic of this video. Let's talk about questions and see how all this is asked in the IMAT. Okay, so this is one of the most interesting part, looking into questions. If you're preparing for the IMAT, you cannot deny this part. Start looking into the last years of the IMAT. If you tell me, oh, I done that, I went back to the last 10 years of the IMAT, I don't know if it's worth it to go more than that. Well, there is a Tokman that is like the IMAT for the Italian. It does not exist anymore. They changed the way they're applying the test, but 2024 has 2023 as well. So it's very valid to go there, download those tests and translate it on Google Translator, because the exams are pretty much the same when they were preparing together. So it's really worth it to do that. I have here the IMAT from 2025, so really recent. I wanted to make a video just talking about this test, but the bottom line here is that it didn't really change, and it's undeniable that Bioenergetics takes a really huge slice of this cake. So let's just take a look through it and see what other questions that we can answer based on what we saw so far. I know the answer is right here, so it's not much as of fun, but the idea is here is not to make like a little mock test. It's just to see the type of questions and the answer they are offering us, okay? First question we have here is very interesting, and we just talked about this. I know this question was considered quite hard for a lot of people. I heard about it. The only thing is that we don't give enough focus and emphasis into the redox reaction. Basically, it's asking us here that during the lactic fermentation, the pyruvate acid is, so the fermentation It's that plan B that we just talked about. The idea is to receive the electrons. And if it's receiving the electrons, the answer here is straightforward. It's going to reduce so the NADH can oxidize. If you have this clear in your mind, you will not be caught up into surprise. Following that, we have question 11 asking which of the following statement about glycolysis is correct. Questions like this that are a little bit more open and then demand you to pay attention, you should do exactly as I said. Pay attention, read carefully, read as many times as you need. Do it like some review of the topic of whatever there is in your mind, like a little brainstorm of the topic, so you can orientate your reading to see and identify what is wrong and what is right. In this case, it's the first option here, and let's see the description. So when a molecule of glucose is converted to pyruvate in glycolysis, 2 NAD plus are reduced. Perfect. So we're talking about glycolysis. Forget about fermentation. We are oxidizing the glucose in order to formate pyruvate. Okay, we're also producing ATP, but also NADH. So having that mantra 2, 2, 2 in mind, 2 pyruvates, 2 ATPs, and 2 NADH would help you a lot to answer it straightforward. I know it's quite easy just to say, oh, this is the correct answer because it's right here, screaming into our eyes. But whenever this is like mixed into the others and you have the pressure, that's when you really need to pay attention. So that's why mock test is really important, and to have questions similar as this one will help you to prepare you for real life. Third question here, 12. This is the third in a row. We did not talk about the Krebs cycle yet, so I will just leave this apart, but it's the same topic of bioenergetics. Our next video will be sharp enough to talk about this. But it's once again, it's a question that requires from you remembering the proportions, the rate of byproducts that are produced in the final reaction. So there's not much a secret here. Next question we have here, it's also about bioenergetics. This one talking about oxidative phosphorylation. So quite an order. They started with fermentation, talking about glycolysis, Krebs cycle, and now oxidative phosphorylation. So they're really focusing bioenergetics. I'll leave this for another video. Then cytochrome, also related to mitochondria in some way. And then they shift a little bit, talking about different subjects, but it doesn't really last long. They're talking about enzymes here, catalysis. Question 20, also talking about enzyme. Don't worry, we're gonna talk all about it. Oxidative phosphorylation once again. So you can see that they are really heavily talking about bioenergetics. This is why this is my first playlist that I decided to record. This is what I'm focusing the most because it's heavily the most questioned topic here in the IMED 2025. And if you go back to 2024, it's just as like that. Okay guys, so I'll leave the next questions for the next videos. If you enjoyed this video, please like it and subscribe to the channel if you're new. This is what really motivates me, and if you want to see more videos like this, just let me know. I have plans to record about anatomy, physiology, biocell, and other subjects as well. So I'll be seeing you all in our next video.